Good morning, everybody. Welcome to the SACS 20th Annual Biotech Europe Forum, or Biotech in Europe Forum. This is the Pandemic Response Day. This is a vaccine panel. Obviously, given the topical nature, we will focus mostly on COVID vaccines, but we have four experts from the vaccine world, sort of throughout the vaccine world. They're nicely placed in different areas. And so we should get a broad discussion on both vaccines and then, of course, how things are progressing but with COVID, but also how new technologies may hit COVID. So I was going to ask each of the panelists to introduce themselves. Andrei, why don't you start and we'll go around the room?
Yes, thank you. I'm Andrei Floroiu, CEO of Vaxart. And should I tell a few things about the company also here?
Very briefly, there's a section for that at the end. So
we are an oral vaccine company, and we have an oral COVID oral vaccine about to enter the clinic.
Perfect. Andrew?
Hi, good morning. My name is Andrew Allen. I'm the president and CEO of Gritstone Oncology, and I'm one of the co-founders of that company. I'm based on the West Coast, Smoky, USA.
Fred?
Good morning, everyone. My name is Frederic Ors. I'm the CEO of IMV, and we are developing a new platform for delivery to the immune system, and we have a peptide-based COVID-19 vaccine about to enter human clinical trials.
Great. And Johan?
Good morning or good afternoon. My name is Johan van Hoof, and I'm leading the R&D organization within Janssen for the vaccine pipeline. Working in this field since 1987, so I'm getting quite old.
Fair enough. Okay, so just as a little bit of intro for people who don't know vaccines, vaccines are hard to make, or they're either very easy or very hard. And I think the best data to suggest that is there are 7 new vaccines, 7 truly novel vaccines in the last 25 years. That stand against about 500 therapeutics. So to make new vaccines is really very difficult. And I think people know there are no vaccines for HIV, for hepatitis C, and things as seemingly as commonplace as the flu are often active less than 50% of the time. The COVID effort has been astoundingly intense. I think part of it is because of the money, but also because of the need. So things have gone at a remarkable speed, and hopefully we'll talk a little bit about where things are and some of the thoughts about moving that fast. I think people know there are about 175 programs. I think probably all of our panels are either fully involved in programs or at least thinking about them. We have at this point 6 leading programs. 5 have kind of been named and 1 lags behind. There are 2 RNA, or lags behind in appreciation. Let me just put it that way. There are 2 RNA approaches. There are several viral approaches, 2 adeno approaches, and then a replicating viral approach from Merck. And then there is really only 1 pure protein approach that is at this level from Sanofi. And then in the background, as I say, there are about 170 programs, most of which have moved a little slower, maybe are focused on what the first programs might have problems with. Most of the first programs use a common antigen. They're quite related. So if it doesn't work, there's an awful lot of things to go on. So that's kind of where we sit. I thought I would start the program by asking Johan, who's at J&J, is very deep in the adeno field. Just your thoughts about the adeno approach to COVID vaccine. What are the advantages? What are the disadvantages? And adeno and mRNA, at least in terms of development, are the furthest ahead. So maybe you could contrast the 2 approaches. And, for instance, Andrew, I know you know both adeno and RNA vaccines well. So as people want to jump in, it's great. But Johan, why don't you start us off?
Sure, thank you. And perhaps I'll focus most on what I know most about, which is indeed the adenovectors that we are using. I think that when you reflect on what critical features should be for platforms that are suitable for COVID vaccines, then I do think vectored vaccines, whether they are non-replicating or replicating, are very well positioned. I think that we certainly the adenovector that we have been working with over the last 10 years, we've learned a lot about it, and we have seen across the different research programs that we really have very robust immune responses, both on the humoral side, but also cell-mediated immunity side. And so from that perspective, back in January, when we heard about this, we felt that this could indeed fit with our platform. The other critical feature is that you really want to have a pure Th1 type of immune response, certainly in light of potential concerns about enhanced disease. And also there, we know from experience that in humans, we have seen always very clear Th1 type of responses. When you go in thousands and millions of people with a vaccine, then it is useful to have a platform which already has quite some extensive clinical experience. And so across the programs that we are running using this platform, we now have vaccinated in our clinical trials more than 100,000 people. And not only in healthy adults, but also in children down to the age of 4 months, also in people over 65, up in their 80s, also pregnant women, HIV positive patients. So from that perspective, we feel that having a solid safety database can also be very useful in this context. Combine this with the fact that we consider this really as a plug-and-play platform, which already has been scaled up at in bioreactor levels of 900 liter, which we combine with our cell line, which can be grown at very high cell density, and that results in high yields and high output. And certainly in the emergency that we're living, having a very fast scale up and producing large amounts of vaccines is certainly also very interesting. So from that perspective, we feel that vector vaccines are actually well established and well suited for this purpose. In terms of comparing it to the RNA platform, and I think we all are really having a fight and we are competing against the virus. I think we are not really competing that much against each other. There will be a need for several vaccines because the needs are enormous. And certainly with RNA viruses, you can go faster into the clinic than you can do with the vector type of viruses, because certainly from our side, you need to go through the steps of preparing master seeds, working seeds, and so on, which is less fast than an RNA vaccine. On the other hand, due to the experience, we've been able then to go fast and also to have fast scale up. So all of that combined makes us very comfortable that indeed a vectored vaccine can actually be very well positioned to be part of the solution here.
Interesting. Andrew, maybe to you, I mean, I think Johan uses a human adenoviral background where your approach has all been with, I think, a non-human primate. And any thoughts there on the platforms? Are they interchangeable?
Yeah, obviously, I agree with everything that Johan said. And we're standing somewhat on his shoulders as we built our platform, which currently is in clinical testing in oncology. Obviously a different application, but the goals were somewhat similar. The goal was to generate particularly a strong CD8 T-cell response. And the data we have from Johan and others' work suggests that the adenovirus is the most potent priming vector that we have for humans to elicit a CD8 T-cell response. You do, of course, get CD4 responses. You get a strong humoral response as well. Frankly, those are fairly straightforward. Most vaccines will generate CD4s and humoral responses, and we're seeing that, within the first generation of the COVID vaccines. CD8s are harder. And so that's why there's been this growing interest, I think, in the adenoviral platform. And different adenoviruses can work. One of the challenges is that adenoviral infections in humans are common. And so there are patient or subjects who have neutralizing anti-adenoviral antibody titers in their blood as a consequence of natural infection that can interfere with the efficacy of a vaccine, because essentially the vaccine gets neutralized by those pre-existing antibodies. And so that's the motivation for moving away from human adenovirus into a non-human, typically a chimpanzee, is that the immunogenicity is preserved, but there is less cross-reactivity with antibodies to the human adenoviruses. such that you have less of this pre-existing immunity problem. So that's the logic for using the chimp adenos. And obviously there's been good data supporting that approach. But otherwise, obviously, our belief is that many of the immunologic mechanisms that drive the utility of this platform and of this vector are pretty consistent between the adenovirus and the chimpanzee adenovirus.
If you allow, I just would like to make a comment indeed on a question that comes up very often is about interference of existing pre-vaccination immune titers against or antibodies against adenovectors. The selection that we have made was well intentional because we really were looking for a vector that has very low seroprevalence. And in addition, when you see people been in contact with adeno26, the levels of antibodies they generate are in general relatively low compared to what you see for adeno5. We actually had the opportunity to study in depth potential interference between antibodies before vaccination and vaccine take. And for adeno26 in the Western population, this seroprevalence is pretty low. However, when you go to Africa, you see that it's present. And there we clearly have seen that there is no interference between these pre-vaccination titers and vaccination, the vaccine take, which probably is related to, on one hand, the relatively low titers in that population, and on the other hand, the relatively high doses of adenovector we give. But certainly it is a point to watch, but we were very happy to see from experience now that we don't see that interference.
Yeah, and we have a little data with COVID, right? With CanSino published their data because they're using a human Ad5.
Yeah,
Yeah, and they showed that obviously the pre-existence of neutralizing antibodies in subjects did have impact on the immunogenicity of that particular vaccine, as Johan said, and that's with Ad5, which is the commonest human adenoviral infection. So Janssen uses typically Ad26, if I'm right, Johan, which obviously is a different serotype. And this has also relevance to the notion of boosting that we may come on to, because you prime with an adenovirus and you will elicit, of course, the response to the cargo, the payload, the thing we're interested in, whether that's spike from SARS-CoV-2 or something else. But you will also elicit an antibody response to the surface proteins, the hexon proteins typically, of the adenovirus as well, such that when you then come in with a boosting dose, you need to be a little bit careful about neutralization of the boost. And so that's the other element of anti-vector immunity that's relevant with adenoviruses. So as is always the case, there's no free lunch here. You know, it's a very, very potent system, but it does come with its own set of issues you have to kind of think about and deal with. They're all manageable, but you just have to deal with them thoughtfully.
And one follow-up for you, Johan. I believe J&J also uses a specific cell line that you've used for some time. How game-changing is that relative to the other adeno approaches in terms of if this works, you're gonna make a lot of it?
That is correct. Yes, we actually, when we look to our Ebola vaccine, for instance, when we produced it back in 2014 in bioreactors of 20 liter, we had an output of about 180,000 doses at that level, which is really remarkable compared to if you would do that on a viral cell or whatever. And we see proportional gains on the yields going into the 900 liters. So it results in, with a relatively low footprint, you actually have very high output. It's a combination of the cell line itself, but also the culture methods that we have developed, because we are culturing these cells at very high cell density. And so paradoxically, it results in a situation where historically your rate limiting step is the volume of your bioreactors and the footprint you need for that. And now the bioreactors are relatively small, it's your downstream that risks to be overwhelmed by your output. So you really need to be vigilant and make sure that you plan for sufficient space for the downstream operations.
Okay, great. So switch gears a little bit, Fred. Proteins, subunits, peptides, they're the workhorses of a lot of the vaccine world, but they're kind of unrepresented on the most advanced list. Maybe you give us a sense of your thoughts on the advantages there, and a little bit and everyone can chime in here. Everyone's worried about antibody exacerbated disease. If you make a lot of spurious, if you get a spurious immune response, how dangerous is it? And we have tiny little bits of data from primates suggesting it's okay. I'd love to hear everybody's view on really how okay is it? When will we really know if we're out of the woods? So maybe Fred, talk a little bit about the protein side of the game.
Okay, well, let me say one thing about your intro, Tom, about the unprecedented level of development that we see in the space, and combination of the money available and the need. But I think that there's something else to me today here, which is really fascinating. I think to be in the industry, in this industry at this moment of time, we are seeing, I believe, a level of innovation in the space that is unlike anything else we've seen in the last 50 years, I would argue. And typically in the pharma world, the cycle of innovation is very long. It takes a long time to bring new platform and new products into market. But what we're seeing with COVID-19 in the vaccine world is a number of new technologies that are being developed and are the leaders, I think, because they are bringing something new to the game, something that's going to be very valuable for the situation. But this situation, I think, a lot of people believe that's kind of just a rehearsal. We're going to be facing more of those in the future. And it's great that, we have a large spectrum of technologies, that are being tested and developed, into clinical trials, including subunit vaccine. In my view, subunit vaccines, I think they are more conventional, I would say. Even though they might not be the first because of the specific of the development of subunit vaccines. Novavax is in the race, I would say. They might not be yet in phase 3, but they have, generated, pretty good data. Some people could argue this is the best data that has been generated in humans, but, honestly, we don't really know a lot about, correlation between, what we are able to measure in the blood and protection. So, I think, everybody's on kind of the same level for now. One thing that, I think it's encouraging with subunits is that, because they've, there are some products on the market and there's some experience around them. I think we can have a better appreciation of what, efficacy, because we've seen it with flu, we've seen it with other products. And the reason why they are using adjuvants actually because based on the experience, with flu and other vaccines, they know that in the elderly population, for example, an adjuvant is something that is required to get, sustained, immunogenicity and protection. So that's why, you're seeing more adjuvants with subunit vaccines. There's also, and I'm going to be talking a little bit of what we are doing, but there's also a number, I think there's about 12, products in preclinical that, are using more targeted, kind of subunit of subunit pieces of the virus, basically peptides, to generate protection. And in this case, it's a bit different. I think it's in my view, the future of vaccination because, this is rational design and this is, improving over what, a natural infection, will give you as protection. It's like, precise medicine for vaccines. The challenge with subunits is that, if you are using very low molecular weight, antigens, you lose immunogenicity. And that's always been the challenge for those technologies and thus, the use of adjuvants. But I think, like for mRNA, there is a growing understanding the delivery vehicle of those subunits, is playing actually a very important role on, the ability for us to generate, a good immune response that's going to be long lasting. And I think that's maybe an area of innovation where, we might see new technologies, coming in the vaccine space and bringing again some precision medicine in that space. The last piece to make the link to the question you ask about, vaccine enhanced disease and the risk that, exist with COVID-19 based on, I guess, the experience with respiratory viruses, and the issue of inflammation in the lungs. It's so far, personally, I'm not aware of, I know there've been a lot of, safety studies, done around that with a large spectrum of different technologies. And to my knowledge, there has not been any report of an effect, whether in HPs or in human or in smaller animal models. So I think, the question will remain, but so far to me, it doesn't look like it's going to be a significant issue down the road.
All right, fantastic. Probably come back to some peptide-related stuff. I have to say, you alluded to what I think is an interesting point. It's one of my favorite fun facts. But all of the coronavirus infections are thought to come from bats, and the epidemiologists think there are at least another 5,000 in bats that we currently have. And it's just a matter of them finding an intermediate species. So this may not be a one-time effort, but let's hope. You can make the argument the current coronavirus kind of has the worst constellation of properties, but it's kind of an interesting that we may be doing this for a while. So, Andrei, you are at some level interested in the route of vaccination, and I think that that's become a fairly topical area in this field, that the lung might be a good place to get innate immunity memory, things like that might be relevant. So maybe you could talk a little bit about what Vaxart does and how it might be relevant. I think you have an oral COVID program, correct?
Yeah, so the company was founded because we believe that vaccines that look like this, I don't know how to show them because I have the virtual background, this is very, very suboptimal. This is like the internal combustion engine. It's noisy, it pollutes, it's slow. It's just, we can do better, right? So we think it should look like this. And of course, as my peers know, it takes a long, long time to get there. So I'm gonna try to explain the two areas in which I think we can really transform the space. First of all, we are also a vector company. We use Ad5. We believe that's very strong and gives good results and has all the advantages that have been talked about previously. But one thing that fits very well COVID-19 is that the pill also triggers mucosal immunity, right? So think about it conceptually. Airborne viruses like flu and COVID, why would you inject against them? Why would you trigger systemic immunity when in fact they infect your mucosa? And now we also know that people that shed most COVID tend to have the most GI symptoms. So we have shown that our oral vaccine triggers very strong mucosal IgAs in the nose, in the mouth, in the lungs, and in the gut. And in January this year was the first time when we were able to show there was a Lancet publication head-to-head against Sanofi's Fluzone that at least in flu, in a phase 2 human study, our vaccine trended positive, showed better results than Sanofi. So this is very encouraging for us. We believe that actually we can confer better immunity against COVID. And you will see in our phase 3 studies that our endpoints are going to be different. We're going to be looking at preventing infection and at preventing transmission, something that we don't think injectable vaccines can do. There have been some signs of that in monkeys, but as our founder says, monkeys are not little people, so it's not a great model for humans, but we'll be shooting to not only show that we can lessen illness, but like in the Lancet paper, that we can have the virus basically bounce off of you. So that's number one. Number two, which is, I think, is becoming more and more important, and at least in the US press, also because the COVID fight has been politicized and you have, lots of people against Trump, lots of people pro-Trump. they started to pay more and more attention to what it takes to get this into people. Okay, so you have it. So let's say Moderna is first over the line. Who has -70 fridges? How do you distribute that? How do you store it? How do you get to the doctor's office? And then how do you inoculate it? So I think people realize that it's going to take many, many months to vaccinate with a needle, whether it's refrigerated or not. This one, you just put it in the mail. In a week, you vaccinate the whole country. Maybe Amazon drones, somebody has suggested to us. So we think that's a huge advantage too. And finally, the third one, look, we are people, right? I personally don't like needles. Some people don't like to interact with others and stand in line. My parents are over 70. They are not looking forward to interacting with a healthcare person or going out to get a vaccine. And because of all these issues that have to do with injectables, many people are just not going to get vaccinated. And I think that's going to impact the success of a mass vaccination campaign, because the goal is to get, over 70% of the people vaccinated. And, I'm going to state the obvious. In developing countries, I think a room temperature stable pill such as ours, if it works, of course, and we think it will, it has huge, huge advantages. So, we may not be the first one there, but we are moving very fast and very aggressively to the clinic, and then afterwards to try to prove the points that I just mentioned.
Thank you. As far as I know, the only company of the companies in deeper trials that are talking this way is Merck, which has used the phrase swish and swallow for their vaccine, for a measles-based vaccine. Do people have opinions on whether that's significantly further away than adeno? Is that ready to go, where it's really the technology's proven, or is that a more distant thing? Does anyone have a sense of those programs?
Yeah, let me say something here, which I think Johan alluded to. Our platform has been in 12 clinical trials against 6 viruses. Unlike Johan's, we're not J&J, so instead of 100,000 patients, we had about 500. But the point is that unlike some of the newer technologies, including mRNA, you have more than a year follow-ups and across various viruses, so you kind of get a sense of the safety and tolerability. I think some of the other programs sound, at least, from what I know, more like research projects, but I don't know too much about them. Maybe my colleagues do.
No takers?
No.
I know people don't like it. Okay, so maybe we can switch to some general questions now that I think are fairly topical. People's thoughts, and I think J&J has talked a fair bit about this, on the importance of a one-shot vaccine, both in terms of getting to people to take it, but also to get a readout from these trials. 3 of the companies have already dosed 25,000 people, and they're not sure yet. And I think one of the reasons is these 2-dose vaccines, you have nearly a month before protection. So maybe you can start, Johan, because you guys talk a lot about one and done, but how big a deal is it in the trial arena? Could it make the trials go much faster? You may have muted yourself. I'm not sure.
You're on mute, Johan.
Yeah, sorry. I think that at the center of the discussion around the design of the phase 3 trials should really be the science and the data that you have in your phase 1 and in your preclinical data. And based on that, you decide whether you need 1 or 2 doses. And that is actually the reason why we feel that a single dose can go there. I think there are 2 aspects that we need to cover, whether you now have a single dose regimen or a 2-dose regimen. At the center of the challenge is that you just are dependent on the incidence of the disease in your study population. And we also know that when you recruit people, there is a certain bias because people who are motivated to participate might actually also become more cautious about making sure they don't get COVID. So I think that one of the reasons why you see the sample sizes going up is that most likely the number of endpoints that you reach in your study are perhaps less than you assumed initially. So I think that is part of the challenge, and also why it's so difficult to predict when you will reach that number of events needed to eventually have your interim analysis and determine to what extent you have protection or not. Of course, indeed, if you can start counting after your first dose, then you do have some gain because you win already several weeks in your race to having the number of events. But the difference is not, there is a difference, but it's not that dramatic. What is true is that, of course, when you look, for instance, to the WHO website, in terms of profile, that the desired profile for COVID vaccines, they actually see potentially two different approaches. One is in an outbreak situation where you really want to go for mass vaccination, where you want to vaccinate hundreds and thousands of people in a very short time period. The preference is really to go for a single dose shot. And eventually even being prepared to trade off in terms of absolute efficacy or in terms of durability. And that is why we think that we actually want to evaluate both schedules. We strongly believe our single shot has potential to be protective, and that is based on the protection data that we get in non-human primates. I'm referring to work that was done in collaboration with Dan Baruch from the Boston area, which was published in Nature a few weeks ago. And based on those data and additional data that we have generated meanwhile, we think there is a high likelihood that a single shot can already be protective. What is unknown is about the durability of that protection. Based on our platform experience, we are cautiously optimistic, at least in our other projects. We do see pretty good persistence of our immune responses, both humoral and cellular, and better than conventionally reported. For instance, when we compare our Zika vaccine with the inactivated Zika vaccine, we do see clearly that we have better immunogenicity, but also better persistence. The decay of the antibodies is different. It's much slower. However, COVID is a virus we don't know that well. Nobody knows exactly how much you need and what the importance is of cell memory, cell-mediated immunity versus the antibodies. And that's why we feel it's important to evaluate both schedules. Also knowing that in an outbreak setting, worldwide vaccination of millions of people, a single dose might actually help you tremendously to get impact on the epidemic.
Yeah, can I add in here, Tom? I think this issue of durability is absolutely key, and it's inconsistent with obviously moving quickly. Because if you vaccinate a large population and show that after 2 months you have protection, well, that obviously is great. That's, we didn't have that. Now we have it. That's excellent. But of course, what happens at month 3 and month 4 and month 6 and so on? And what is clear is that the decay of the humoral response with SARS-CoV-2, based on a nice paper in the New England Journal, is pretty fast. And of course, that's expected, right? Immune responses, they wax and they wane. That is how they normally behave. And so the unknown is what threshold do I need for protection? And generically, we believe that humoral immunity is generally more transient than cell-mediated immunity. CD8 memory seems to be somewhat more persistent. And there's a reason, of course, our natural immune response uses both antibodies and T cells, because both they complement each other. They're different arms of the immune response. And so we may have a good enough vaccine that focuses on the humoral arm, but it is possible, maybe probable, that a better vaccine will generate both humoral and cell-mediated immunity, and the memory associated with that cell-mediated immunity may be very important for durable protection. So one's hope is that as we start to analyze data coming off of the big phase 3s currently running, the data will be broken down by time course. What was your relative risk of infection 1 month after treatment, 3 months after treatment, 6 months after treatment? And this, of course, is likely to guide the necessary boosting schedule, because as we all know, most vaccines that are currently used require boosts. And the periodicity of those boosts should be determined empirically, ideally. And again, what we've seen is that single dose, for example, of the RNAs looks to be insufficient, which is why both of the leading RNA players, the mRNA players, are using boost intervals of 3 or 4 weeks. And that obviously then seems to be sufficient. And in non-human primates, if you study them, 4 or 5 weeks after initial vaccination, you get protection. So again, we're kind of moving the ball. But how durable is that protection? We haven't seen primate studies done 3 months after prime, for example, 6 months after prime. I think Johan's alluding to that, obviously, that's the kind of experiment that you want to see. You want to see monkeys that were vaccinated 6 months ago now being challenged. And do we see the same degree of protection that we observed 1 month after initial vaccination? So this durability issue is absolutely critical for determining the optimal vaccine and the optimal vaccine regimen, as Johan said. Now, maybe transient protection will help because it will suppress the pandemic to some extent, but it's not like the whole world is going to be simultaneously vaccinated on the same day. And so I think the utility of that sort of transient protection may be relatively modest, given the likely kind of staggered rollout across countries and across the world. So this is just a challenge we're all going to have to embrace. But durability is going to be a critical determinant, I think, of long-term success here.
Just to follow up, because I think you're kind of the T-cell guy on the panel. If in fact a strong CD8 response is needed, are we almost inherently looking at multiple vaccinations? Is it possible to get a strong, durable T-cell memory response, CD8 T-cell memory response with a brief administration?
It is possible in principle, yes, because that's what happens with natural infections, right? We often get a single infection and we can have lifelong immunity to that infection. So in principle, if our vaccines are good enough, then yes, that's achievable. It's obviously going to be easier if we prime and then we boost. So that's more likely, I think, in the near term. But this notion of CD8 immunity is obviously important. And you have to think about the issue of antigens. This obviously becomes relevant as you think about long-term protection, is this notion of mutation and mutagenicity. And obviously we see this with flu, right? Flu is the classic example where we focus on these surface proteins, the hemagglutinin, neuraminidase, they change a lot, and therefore vaccination in 1 year provides limited protection the second year. And so you have to then do a second, another vaccination with a different set of these surface proteins because they're evolving so fast. Now, the good news, it seems to be, is that within the family of the coronavirus, mutational rate is pretty low. They have a high fidelity proofreading enzymatic function encoded within the viral genome, so that mutability is actually quite low. Although, of course, under selection pressure, we may be changing that to kinetic. So then the issue becomes, well, which antigens do you want? And this obviously is a huge topic where a lot of the current focus right now is on spike. And spike makes sense because neutralizing antibodies to spike provide functional neutralization of infection. So that's the clear logic. However, it is likely that spike will be one of the first proteins to evolve as SARS-CoV-3 comes along. And if you look back at the 3 epidemics/pandemics we've had from coronavirus, so we had SARS back in 2002, and MERS in 2012, COVID in 2019, obviously spike changes significantly, particularly with MERS. And so there's, as far as we can tell, really very little, if any, cross-reactivity between the spike of these different variants of the virus. But other proteins, some of the intracellular or intraviral, I should say, proteins, are much more conserved evolutionarily. And so the sweet spot would be to find evolutionarily conserved epitopes, which or segments of the protein which actually function as T-cell epitopes. And that's obviously work that's ongoing. And Alex Setti has been publishing, a paper just came out this week, again, showing this persistence of certain CD8 epitopes characterized within the viral genome that you see reproducibly between humans. And Oxford just published a paper a couple of weeks ago on the same topic. So we're starting to converge, I think, on this notion that CD8 epitopes outside of spike exist. They're potentially very important. And that's the kind of an area of interest, I think, as we think about durable protection, both against SARS-CoV-2, but also, to your point about bats and this big reservoir of the next coronavirus, SARS-CoV-3. Immunity against that is unlikely, I think, to be driven through spike-based immunity. It's more likely to be one of these more conserved, perhaps, structural or even non-structural proteins within the viral genome.
That's a perfect segue, Fred, because you guys have a very different approach. So rather than necessarily, I'm sure you are generating T cells, but you are in fact not inoculating multiple times. You are inoculating over a very extended period. So maybe you can talk about that aspect of your program and how you think it plays into this question.
Sure. Well, I actually agree with the previous comments that, on the long run, I think that it will be duration of protection that, will be a very important factor. If you look, up to now, the focus has been very much on peak responses. And I'm making my career based on area under the curve, for oncology and also for COVID-19. So yeah, what we believe in is that the duration of the exposure of the antigen to the immune system is a key factor in the magnitude, the duration, and the quality of the immune response that you generate against an infection, and drives the quality of the protection you're going to have from a vaccine. And actually, there's some very interesting papers on people who got COVID and the difference you see in the immune response between someone that was asymptomatic and someone that had a very severe disease. And some of the papers highlight how much better is the immune response when you got it bad, and how much short-lived it could be if you're asymptomatic, and the chances you could get it after. The CD8 could play a different role, and we don't have all the data, so I think more will come. But generally speaking, with infections, if you get it bad, you're better protected, and so that's for us, the technology that we have, is actually sustaining the delivery over a very extended period of time. When with mRNA vaccination, I'll take that because the example was given. I think they say it's 48 hours and the bolus injection is all gone. That's kind of a safety feature of the mRNA, but that's also, in my view, the challenge of mRNA vaccines, that the time of exposure is very short, and you wonder what kind of duration and quality of protection can be generated from such a short exposure into the immune system. For us, in comparison, we have weeks and months of exposure to the antigen, generated by the formulation. We've mainly applied it, I have to say, in oncology, and I know you're going to talk about that in a few minutes. But for us, to emphasize what Andrew said, about CD8, the ability, to maintain, CD8 in the blood of patients, we believe is correlated with the exposure. And what we've been doing in oncology is, because of the way, we can deliver antigens, we have the ability to repeat, injection over and over, which, it's not possible with vaccines, but in oncology it's possible. We've done it, as an example, with ovarian cancer patient, woman who received the treatment, every 2 months for 3 years. And during that period of 3 years, we have been able to maintain, very high level of CD8, T-cell in the blood, and with clinical benefits, a partial regression that was maintained for more than 3 years, which is, amazing for late stage metastatic ovarian cancer patients. So we do believe, and I do believe that, as for the route of administration, to Andrei's point, the way you deliver the antigen and the time, you expose the antigen to the immune system is also, an important factor into the quality of the protection and the quality of the immune response, you can generate. The good news is that, like I said at the beginning, all of those technologies, are being, tested, and they will go into clinical trials. And, personally, I'm very excited, about the opportunity, to be able to show the differences, between these technology head to head in the same, with the same virus, in a large number of patients. I think, science will make, the science of vaccines, will make amazing progress, in the next, 6 to 12 months in understanding how we can build, better vaccines for the future.
And Andrei, I give you a chance. I mean, given you have a new technology, is persistence of response one of the key deliverables you're focused on? This mucosal?
It is, to what Andrew said, first of all, we worry about the potential that this virus can mutate, and when we designed our antigen, it's actually S and N, and we just published some animal data showing that at least for us, S plus N work much better, including Tcell responses than the S alone. And with our norovirus vaccine, we get this asked all the time, our IgA is lasting, and we know that they last at least through 6 months. So that's encouraging, and we're gonna look at whether they last longer than that. But what I agree with Johan and Andrei, and I think Fred too, is it seems like people and politicians have been focusing on just let's get the first vaccine instead of let's find a solution, right? So people commented about the if you have 2 shots, the logistical complexity to keep track who got what and to make sure they get the second one, the same one, not another one. It's only adds to the problem, right? So my colleagues or peers spoke about the schedule of that we're trying. We're also trying. We believe that 1 pill in our case, not shot, will work, but maybe in the elderly you need 2, and it's just easier to give people 2 pills than to get them back in for another shot. So we are kind of indifferent as to whether 1 pill versus 2 work, but we are confident that 1 should do the trick. And we think that we look at this as a vaccine that would need annual boosting. And I know there is dispute here, particularly with Janssen, which actually is a partner of ours. We believe that in the gut, AD5, actually, we know that doesn't trigger any anti-vector immunity. So for us, it's actually very easy to re-administer the vaccine. Whereas in systemic administration, I think people have shown that you get some anti-vector immunity. I know that Janssen says that they haven't seen that in their vaccine. At least that's what I hear from my scientific colleagues. But we'll see that in the clinic.
All right, great. I wanted to switch gears a little bit. How good does the vaccine have to be? As you talk about efficacy, people choose their words very carefully. I was quite struck at Pfizer's recent event where they really said the preclinical data suggests our vaccine will be 60% effective or better. And Moderna uses the number 60% as well. Is that likely to be how good this is? Do people know from the preclinical data? How good are the preclinical data? I've probably seen more of your preclinical data than anyone else's, Andrew, but just maybe Johan, your sense is, do people have a sense of how good this vaccine is likely to be, or is it totally unknown?
Personally, I think it is unknown. It's very difficult to put a number on it because you don't know how predictive these animal models are for the human situation. The good news is that at least in animals you can protect. The hamster model, which seems to be a good disease model, you do see that you can really prevent the disease, the symptoms. In non-human primates, that illness is less pronounced. It's more mild symptoms. They clear the virus themselves, but you do see that you can prevent actually infection in the animals. So for me, this is sufficient to hope indeed that these type of immune responses will offer a degree of protection in humans. We know that for respiratory infections, it is more difficult to protect against an upper respiratory tract than lower respiratory tract. And lower respiratory tract infection is probably the ones which provokes most disease. So I do think that the type of protection it's going to offer can go from the holy grail, which is preventing infection, sterilizing immunity, to preventing severe disease, preventing hospitalization, intensive care, and death. And I think the data will tell us where we end up. But certainly we have done, for instance, in RSV, a human challenge trial that's in the public domain, and you do see that protection against pure infection, PCR-confirmed infection, was around 50%. When you look, however, to the clinical scores, and of course, in such a human challenge trial, you're careful, you don't want to your people to end up in the clinic. But certainly when you look to the symptom scores, you do see that you actually got up to 85% if you have some more with regards to a more severe type of symptoms. And that is exactly, I think, why the endpoints now are really focusing on preventing disease. And the secondary endpoint is preventing infection. And let's hope indeed that also in the secondary endpoint we see this. But we should not underestimate that preventing respiratory infection is quite challenging. It's possible, but I think we already would be very happy that we do see high efficacy against moderate and severe COVID. Can we have more? I think the data will tell us. I think no one can really predict it today.
Yeah, I think that's right, Johan. The Moderna protocol, the primary endpoint is prevention of infection, and the secondary is severe disease. And you could argue that's kind of upside down, because obviously what we care about is severe disease. If we were able to convert this to the common cold, I think obviously that would be success. That would be therapeutic success. And that really is what we care about. And the question, I think, is how do you get to prevention of severe disease? Is the path to that to have complete population immunity, sterilizing immunity, so there is no infection out there? Or is it to simply attenuate disease? And those may end up actually being rather different goals. And I think a little bit is going to be driven by the biology, because what we've learned is that, as you know, for the majority of subjects who get infected, the disease is relatively minor. And it actually seems to be the immunopathologic response to the virus that causes severe disease. Now, will we attenuate that? If you have minor antibody levels, for example, would that convert you from being a subject who was going to get really sick to somebody who just gets minor disease? Or because it's actually immunopathology driven, does a low level of antibodies actually not really change that? And so if you get infected, you're still going to get severe disease. That's kind of a central question. And we just don't have the answers. And the human data obviously will be the only way to resolve that. So we need to understand this issue of degree of prevention of all infections, but obviously perhaps most importantly, prevention of severe disease. And there you just need numbers. And again, we talked about durability. I think this is the challenge of making very rapid vaccination approval decisions on very limited data, which may be actually comprising, in large part, minor infection, relatively asymptomatic or minimally symptomatic infections driving an approval, when what you care about is actually a different endpoint, which is prevention of severe disease.
That's why I think it was very useful just to tackle that aspect. That FDA recently issued a guidance that at least in your efficacy studies, you should have demonstrated that you prevent also against severe disease. So they want to have at least 5 severe cases in the endpoints. And I think that is actually to address really the concern that you raise. With regard to preventing disease or getting less severe disease, I do think that we should not underestimate the role of cell-mediated immunity, because that's, I think, where especially also CD8 responses hopefully can help us to achieve that.
And I would like to add perhaps another third endpoint, which is prevention of transmission, right? That's why we wear masks from a societal point of view. Maybe I'm infected, I don't want to infect you. And I think from a practical point of view, I can go into with my parents, I'm less concerned whether I get infected, and I'm a lot more concerned to infect them. So I think if you can show that you prevent infection, that's another way to actually tackle the epidemic. And we've seen in other viruses, for example, it's shown that in norovirus, you actually want to protect the kids because they are the ones that end up transmitting the disease, and you flatten the curve that way. Probably in flu, it's similar. So we'll see. I think the jury is still out as to how to best fight this.
Fred, do you have a closing comment? We're pretty much out of time. You're muted.
Well, I think Johan alluded to the RSV case, and I think that's a virus we should look at in terms of severe disease, because it could be different with COVID-19, but I think there are some evidence that it's a question of viral load and whether the virus get a chance to get deep in your lungs. And then, you have this immunopathology starting from there. But if you have a vaccine that has the capacity, to block this progression, within the respiratory tract, very likely that, you will be able to alleviate, a lot of the severe diseases, and especially in the elderly population, as you know, for RSV, we always talk about, infants, but there's also a lot of cases every year in the elderly population, and they get hospitalized when the virus, progress into the deep lungs and create those type of immunopathology. And I think, for COVID-19, it might be something severe. And I think some vaccines, could actually be effective in blocking that, in the upper respiratory tract, and, making it a call, basically. That's not too bad. So we'll see.
All right, I'm afraid we're out of time. I wanted to thank you all for doing this on the fly. I know it's not easy, so thank you for sharing your thoughts. And the good news is we should know something in November, so we're all hopeful. So thank you very much, and have a good, safe day, and hope you can see the blue sky where you are.